In this module of the sterilization, we will discuss the source of contamination. As concerns the fermentation process, as we have seen in our previous module, that fermentation process is actually the mass culturing that require a suitable medium. So during the fermentation process, when we see the whole organization of the fermentation process, that's first one is the source of contamination is the leakage or the fermentation vessel. Second source of the contamination is actually the medium. When we formulate the medium, when we make the medium and then feed into the fermentation vessel, then the contamination can happen. Then third is when we sparge the medium with the air, then the contamination may come through the air. So there are the number of factors which can be the source of contamination. If we, first of all, if we avoid those and we can have a control check over all those factors from where the contamination can occur, then we can run our fermentation only with our production organism, which we called as our fermentation organism. So how we can avoid the contamination? As concerned the avoidance of the contamination, so the first is we use pure inoculum in the start of the fermentation. If there will be a contamination in our inoculum, then there is no more way by which we can avoid our contamination. Our inoculum should be 100% pure and without any sort of contamination. And second is the medium which we are using and we are employed, because there are two techniques. Either we sterilize the medium inside the fermenter, either we first having the sterilization of the medium apart from the fermentation vessel and then we add into the vessel. So if we employed the medium, then our medium should be sterilized. So the sterilized medium should be employed. In this case, when we are adding the medium sterilized, then our fermenter vessel should be clean and sterilized. So in fourth case, the sterilization of all the material to be added into the fermentation that should be sterilized. What are those materials? That can be antifoam, that can be any other alkali or the acid medium, or if we are running with the fat batch, then those ingredient and those part of the medium which we are adding through the fermentation process, that should be properly sterilized. And then the fifth one, the maintaining the aseptic conditions during the fermentation process, then we can avoid the contamination in our fermentation process. So as concerned, the extent to which the procedures are to be adopted for the avoidance of the contamination, that will help to chalk out the probability of the contamination. If we have a very controlled mechanism of the avoidance of the contamination, then there will be a less probability of the contamination. Why we are talking about the probability of contamination? Because when we say that the number of unwanted microorganisms present into any object, then our whole sterilization process will be. So we can say on the base of the fermentations in two categories, either that was protected or unprotected. So in first case, in most of the fermentation, they are termed as the protected. If the media may be utilized only a limited range of the microorganism, which we called as the process organism, and in the result of the development of a selective growth conditions for our desired organisms in terms of pH, in terms of nutrients, in terms of other conditions. So in case of, just an example, if the breeding of the beer falls into the category, if we are using the hope raisins, tend to inhibit the growth of many microorganisms, and the growth of brewing yeast tend to decrease the pH of the medium. So this is the case when such type, when there is a microorganism, then how that effect on the pH of the medium. So thus the brewing wort are boiled but not necessarily sterilized, and the fermentations are thoroughly cleaned with disinfectants but are not properly sterilized. So also there are some precautions used when the development of the inoculum. So in case of the inoculum development, then that should be having a very pure and having no contamination. So the majority of the fermentations are the protected, but we can say that then there are the majority of the fermentation which we cannot say that they are protected. So if there is a contamination, that can lead to different consequences as we have discussed in our previous modules. So in case of the protected, then we can assure our fermentation process in a positive way, but if our fermentation process is not protected, then so many consequences which we have discussed that can happen.